Hello guys, I am Shubham from ShubhVed Tutorials. This video is about base excision repair. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. So we are discussing about repair pathways, and today we are discussing base excision repair. So this is for the damaged bases. Damaged bases. So we know the damage because of reactions, right? We have discussed in the DNA damage video on DNA damage and replication errors. Uh, those reactions are deamination, methylation, oxidation, right? All these give rise to damaged bases. Okay, so this is done by the enzyme known as glycosylase. Glycosylase. So glycosylase breaks the glycosidic bond. Okay, breaks the glycosidic bond. We can understand from the name glycosylase, right? So this enzyme is very specific according to the DNA damage. And in humans, there are 11 types. In humans, 11 types of glycosylase are present, right? So let's see the pathway now, what exactly it does. So here we can see a sugar phosphate backbone, right? This is a sugar phosphate backbone from 5 prime to 3 prime. This is a sugar and this is a phosphate. Okay, as well as here. And these are the bases, normal bases. And we can see here uracil. See, this we are seeing an example of uracil glycosylase. Okay, the uracil which is not found in DNA, this has to be removed. Why uracil is found? Because of deamination of cytosine. So uracil is present. Now uracil has to be removed. Okay. So here comes uracil glycosylase. Right? So these are the bases. You can see here. So this is a DNA basically. Then glycosylase will cleave here. Glycosylase will cleave here. Okay. After cleavage, the uracil will be removed out. See, this is a free uracil. Uracil is removed out. Then what happens? Uracil is a nucleotide, right? Basically a pyrimidine. Okay, this is a pyrimidine. So here formation of AP site takes place. After glycosylase reaction, what happens? AP site. AP is what? Apurinic or apyrimidic site, right? So this pyrimidine has been removed out, uracil. So here we can see no uracil is present. So this is called AP site. Then the endonucleus and exonucleus, which will act on this AP site. AP means apurinic or apyrimidic site will come. The endonucleus or exonucleus which act on AP site will come, and then the endonucleus will cleave here and make 3' OH. For Pol 1, see we are discussing about prokaryotes. Okay, Pol 1. Pol 1 is for repair, DNA repair. So AP will cleave here. So this will be a 3' OH. Okay, 3' OH. And exonucleus will cut here. Here. So this is a 5' end. This has to be remain intact. Okay, so 5' and 3' cut has been introduced. And then this will be like this. Suppose we can say this is the strand. Here is the damaged base. Okay. First of all, glycosylase will remove the base. Okay. Only this bond will be there. The base has been removed. Right. This is making a AP site. Okay, and then AP endo-exo will do. This is a DNA which breaks. It has been broken down. Then again, this will be released out. Okay, there will be gap, and this will be filled by DNA call 1 in prokaryotes, right? And here is the correct nucleotide now. Okay, there are other nucleotides too, do note, but we are considering here only the correct one. Right, this will be the repair pathway. Okay, got it. So here we saw the example of uracil, right? Removal of uracil from DNA. Right. Also, the other type of damage is repaired by base excision repair are oxo-G. Right, oxo-G is the oxidation product of guanine, which is very harmful. Right, so this has to be removed. Okay, same glycosylase, which is specific for this oxo-G, will come and remove it. Right, and same other than AP endo-exo will come, break the backbone of DNA, and then it will be repaired by the DNA polymerase 1 in the eukaryotes. Okay, now in oxo-G, basically it is in humans. So if the removal does not take place, so what happens? There is another glycosylase which is known as fail-safe glycosylase. Fail-safe glycosylase. What it does? Say oxo-G pairs with adenine. Right. So what this glycosylase will do? It will remove the adenine. It will remove the adenine and incorporate correct nucleotide. That is C, which pairs with G. Okay, but oxo-G is still there. We can see oxo-G is still there. Means damage is still there. Right. So this is fail safe. It is just correcting the complementary nucleotide, not the exact damage phase. Right. It is removing the complementary nucleotide. Incorrect pad. Right. Hence here adenine is there, which has to be removed because G pairs with C. So it is incorporating C instead of A. Got it? So this is fail safe. There is one more example in this fail safe, that is TG pairing. Okay, so removal of T by this fail safe glycosylase. It will remove T, T will be removed, and C will come here. Got it? So this is also seen. We know T, why T has to be incorporated? Deamination of methyl cytosine produces thiamine. Alright? In eukaryotes, the process is same. See, first of all, glycosylase. Then there will be a AP site. See, nucleotide has been removed. Okay? But the backbone is intact. Then incision by AP, endonuclease 1. Okay? What it will do? See, this is a long pathway. There are two pathways, long pathway and short patch pathway. So this is same, failure, AP1. Then neat translation means what? This has to be cut and the repair has been done. See, by Paul delta or epsilon. Okay, Paul delta epsilon incorporates correct nucleotide here. Right, and then strand that was present before is flipped upwards. See, we can see here from the picture, as the strand is synthesized, the previous strand is flipped. See, and then will come endonuclease FEN1, which will cut this strand. And here the join. Okay, joining will take place. The previous strand has been cut, and here the joining, here there is a gap. This gap is sealed by the ligase. Gap is sealed by the ligase. Right? See, when we discuss DNA polymerase, what it does, it will synthesize the strand, but there will be a little gap in the end. Okay, this is to be filled by DNA ligase. Joining, you can say joining, complete joining is done by DNA ligase. Then the strand is continuous. Okay, so this is a long patch pathway. Now in the short pathway, first come lys. Okay, lys is, see, there is a cut here. Then it will replace the nucleotide with the help of AP endonuclease and polymerase beta. Polymerase beta is for base excision repair, right? So here polymerase beta, and then see, this is more direct, we can see, and this is indirect. And after correct incorporation of nucleotide, the gap will be sealed by the ligase. Here ligase is XRCC1 and ligase 3. Okay, XRCC1, ligase 3. So we can say that this is a direct pathway, and this is indirect. Okay, long pathway and short pathway in eukaryotes. Here first glycosylase, which is common enzyme, right? It will do the AP site formation. The purine or pyrimidine is removed, which is damaged basically. Then there is which enzyme? AP endo, right? Endonuclease. Then polymerase delta, epsilon, and in the short pathway beta, polymerase beta, and then ligase. Ligase XRCC1, ligase 1, ligase 3. Right? Ligase 1 is in long pathway, XRCC1, ligase 3 are in the short pathway. Okay, glycosylase enzyme is important, which will recognize the damaged base and remove it from the DNA. Right? So this is about base excision repair. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section.